Okay, good morning, everybody. We left off kind of about a third of the way, maybe through this chapter. We talked about a handful of ways initially that we think about trying to maybe combat organisms with either inanimate objects or on ourselves. And then we shifted over and thought a little bit about some cold situations. And now we want to think heat. So there are lots of ideas, and we showed an autoclave last time talking about some of those potentials, and a pressure cooker is a similar sort of an idea as an autoclave, basically providing an opportunity to get the temperature well above the boiling point through pressurizing a chamber, in this case, the pressure cooker. So lots of home canning, food preservation stuff truly requires that you utilize a pressure cooker to get the temperature up there high enough to kill potential Clostridium spores that may have found their way onto some sort of food stuff. There are all sorts of maybe differences dependent upon the types of things you might be preserving. Green beans, oftentimes a good one to think about for possibly coming in contact with these organisms. So Clostridium, there's all sorts of different species, but botulinum is the one we really think about as a potential for some foodborne illness. It is an anaerobic organism, so we would expect to see these guys in the soil, likely, where oxygen levels are very low, and that lends to these guys being able to grow. When situations are maybe not so good, they start to form spores, and oftentimes it might be the spore that we're actually coming in contact with. A cousin, Clostridium tetani, causes tetanus. We'll go through and talk about the disease production with both of these a little later on. But we think about the spores that we're coming in contact with, or maybe getting inoculated into our body by stepping on a rusty nail. You always hear, oh, a rusty nail, right? Something, puncture wound that may inoculate that into you. In the case of these guys, the organisms could be, the spores from these organisms could be something that finds their way onto foodstuffs. And if they're not preserved properly, the idea would be, through canning with a simple hot water bath. So we bring it up to a boil, but we're not getting above 100 degrees Celsius. Those spores can survive that. They can persist. And as that food stuff cools down, we have sealed it in our jar. We've created an anaerobic environment, and those spores find that to be a hospitable environment. As the temperature drops back down, they germinate, exfoliate, and start growing as the bacteria. Those bacteria in the anaerobic environment start releasing toxins, and it's the toxins in this case that truly make you ill, all right? So through pressure cooking, it helps to ensure that the temperature gets up there enough to kill off these spores. And in this first image, I think it's an awesome example of Clostridium, where we've got, and this isn't a spore stain, but we can see the details really quite well. So a vegetative cell is when it's just minding its own business, being a bacterium, all right? Pulling in nutrients, growing, replicating, and all of these that are all dark, we would classify them as vegetative cells. Then maybe the condition starts to get a little unfavorable, maybe some hints of oxygen, maybe temperature, pH, whatever it might be with some of these bacteria. And in this case, with our Clostridia, the anaerobes, oxygen could be a good stimulus for this. We can start seeing some of these cells going through the process of forming spores. We talked about spore formation a handful of chapters back and showed a little bit of what's going on with that. Here you can see these cells that are starting to form spores within them. And there's definitely some variation with the spore forming microbes as far as where those spores form in the cell and what it might be doing to that bacterial cell. Sometimes, and these slightly swollen, if you will, it's a little bit thicker than the vegetative cell, but not considerable. Clostridium tetani is much more pronounced, and it kind of looks tennis racket-like with a big fat spore on one end, a terminal end. All right. There are others that go more toward the middle. And again, swelling or not swelling, some of these things may help to give us a hint about what we might be looking at as we see them under the microscope. And then free floating. And this one's, got quite a few of them. These are individual spores. The vegetative cells are all gone, and these basically just laying in wait until conditions are favorable again. So we've got a good example of all three different steps that we might be thinking about with some of these spore formers. And remember, for the purposes of our stuff in here, we think about the Clostridium as the anaerobic bacteria, gram-positive rods, but anaerobic bacteria that are capable of forming spores. There's a boatload of different species of them. And then the aerobic gram-positive rods, bacillus, are the spore formers that we think about, both of which have numerous habitats in the environment that they may call home. And they also set the stage for some potential disease conditions within us, right? And in both circumstances, with food preservation, we think about some of these mechanisms that truly can kill these spores, right? When we talked the autoclave and we kind of wrapped up the lecture last time with a little of that, it's some of these spores that can be in those little capsules that you would throw in the autoclave. And when it comes out of the autoclave, you snap it, the liquid media interacts with that spore. If the spore didn't get heated up to the right temperature, it starts to grow and you get a color change in that little ampule. If it did kill it, it stays colorless or whatever it started off with. I believe it's colorless. So that's a good mechanism for that. Sometimes we think about other ways for preservation. So we could think about some of the osmotic pressure, like how much solutes are we thinking about? And that ultimately sets the stage for our bacterial cells to not be able to tolerate these conditions. At least the bulk of them can't tolerate this stuff. So here we've got a raisin as an example, and we've got some of them right here. A hypertonic environment where you've got considerably more solutes on the outside, right, in the solution compared to our cell as our example, or our food products here. It causes the moisture, the liquid to come out of there, causes it to basically lose a lot of the ability for bacteria to say this is suitable, all right? There are some other potentials. Some fungi may look at some of these things and still find it to be a little bit suitable. So it kind of depends on some of the circumstances as far as the way it's being preserved. And sometimes meats might be more of a culprit. maybe similarly with some jams, and it also depends on how these things might be preserved, right? Oftentimes with a jam, you're not using a pressure cooker to deal with that canning process. If you're going to actually can it, the sugar concentrations in there are enough to make it inhibitory. You heat it up, your jam, whatever you're making, it boils, and then it goes into a boiling water bath, and that allows these jars to seal up, but we're not pressure cooking, all right? There are other techniques where we're not going through the water bath situation. You make some freezer jam. That stuff isn't quite as preserved as the one that's gone through the water bath. You have to keep it in the freezer till you're ready to use it, right? And the others can be on the shelf, right? So truly depends on the circumstances with this dehydrating to remove water. In these circumstances, using salt as another mechanism, or boatloads of sugar to lend to this higher hypertonic situation, a higher solute that pulls that. If we could think of this raisin as a bacterium, this would be what's happening to it as well, and it just can't survive that type of situation. So shifting it up a little bit, there are other ways that we may think about targeting microbes. UV radiation, and we showed a little of this when we were thinking some of the genetic stuff. It has the potential to cause some pretty serious damage in the DNA, and dependent upon the length of exposure, that damage can be irreparable. It's going to cause the demise of those microbes. So it causes bonding between adjacent thymines on the same strand, thymine dimers. There are, and we discussed this before, some repair mechanisms that may lend to correcting these problems, but typically lengthy exposures to this makes it so that it doesn't matter. It's a lethal situation. So we can see one of these fume hoods that has one of these lamps. In the biology department, there are a couple portable UV lamps that they can wheel into some labs and use for some decontamination. Really thinking more like microbiology labs, where sometimes you find a bacterium that's maybe become a contaminant, and usually it's some sort of bacillus, some sort of spore former that can persist and then winds up being a contaminant, right? As it gets in there and starts growing, it becomes a problem with whatever it is you're trying to work with. So they can wheel these lamps into the lab. They're on a timer. So it typically goes for about 3 hours, and it's a big, like a big fluorescent tube. These big lamps wheeled in and kind of UV the whole area for the most part. That's the idea, all right? And bringing it to more, sterile sort of a situation, so that the bacillus or whatever your potential contaminant may have been isn't an issue. I remember this was many years ago. There was a lab where they were, I think it was probably comparative anatomy. So they do lots of different dissections. You look at all sorts of animals and comparative anatomy. We're comparing those things to ourselves and thinking about different structures. And it's oftentimes pretty easy to be making these comparisons. And maybe you're doing some of this stuff shortly after you've had actual anatomy class. So you've had an opportunity to see the donors, you've had the ability to go through some of that stuff, and then we step it up and start thinking comparative and making some of these connections. Maybe you flip it around and do it the opposite way, all right? So one of the animals that they deal with oftentimes are some cats. I technically have a handful of stories with this, but one that I recall firsthand, they had a handful of these cats that they were going to dissect, and the TA opened up the bag, and all of these cats were moldy. And that's disturbing because they should be pretty much preserved in some formaldehyde or whatever they're preserving them in. They shouldn't be moldy, right? But they were, right? And they closed the bag up and said, they're like, oh, we found all of these moldy cats. Are you interested in looking at this stuff? I'm like, heck no, I'm not. I don't know what that mold is. And if it's growing on a preserved cat like that, that seems like it's a potential danger, right? I don't want to be exposing myself to that. I think that bag should go to the autoclave, and you should maybe decontaminate, which is what they did. And the light went in there and they decontaminated. My other story, this was from one of the professors way back in the day, that was talking about when he was a student, and the lab manual, when it got time for you to do the cat dissection, you took that upon yourself and you collected yourself a cat, typically from the alley, because cats were just running all over the place, right? Apparently, right? And you would just get one, kill it, skin it, and bring it in. And I was like, what the hell? That would be a lab I would not want to take, right? They have since passed all sorts of different rules and regulations, and you're not doing that type of stuff, right? I'm like, that's the things that drives people away, I would think, right? And if not, you're like, huh, that's kind of weird, right? You just spend your time killing cats. There's maybe some sort of problems with that. I start to wonder, right? Serial killer like stuff is what I'm thinking, right? It just makes you wonder, right? So they don't do that stuff anymore. There's still dissections, but not collecting your own things, right? I remember a parasitology class that I took. We weren't killing things, but we did a handful of different dissections, and some of the stuff was roadkill, something scooped up right off the side of the road, brought in, and that's pretty ripe. Brought in, and then we would go to town digging through there trying to see what sorts of things might be in this intestine of this animal, and you probably ought to guarantee you're going to find stuff, right? Kind of crazy, but that I felt a little better about that, right? And I also didn't have to go scoop up any roadkill, but for the love of science, right? So I don't know how we got off on this UV tangent like that, but there you go. Fun stories for 9 in the morning. So radiation, another potential for dealing with things. Produce could be a good one. We mentioned a little bit with that pasteurization, maybe milk could be irradiated as well. So as these move through this chamber, basically they're getting some gamma radiation. They're getting a dose of radiation that basically ensures anything that was on there has been killed. There are some other potentials with this, because typically when they go through the gamma radiation, the seeds within there no longer are going to be viable. So for some of those circumstances, maybe that's good for these producers, because you can't just take this and plant it and expect to grow more of whatever we got going on, apples in this case, right? So there are different mechanisms that they will stamp onto things that show if they've been dealt with in those fashions. And it really kind of depends on circumstances and where you're at. So we showed that HEPA filter or the fume hood, and I mentioned the HEPA filter. I'm showing you a HEPA filter right now. I mentioned those hoods don't just pipe the air right to the outside. They go through some levels of filtration to ensure that what's really being released out has been treated or dealt with. All right. And so these HEPA filters look furnace filter like, but big. All right. Typically very thick filters that have lots of different levels of porosity as we move through there. That the idea would be things as they're moving through find themselves trapped somewhere within these filters, all right? Making it so that the air that moves through there has been rendered safe, all right? So all sorts of different potentials as we're moving through and trapping those within there. So that's the idea. And sometimes maybe a handful of layers thick. There are other ideas that we might potentially think about smaller scale, all right? And maybe for dealing with laboratory solutions that can't tolerate going through an autoclave. Those high temperatures might destroy it, all right? Are you guys doing biochemical tests? You doing that stuff, the sugar ferments and all of those things? Do you see the little urea broths, little teeny tubes? You know what I'm talking about? Kind of peach colored, little bit of liquid in there. No? Yes? Couple people saying yes. Some other people are like, I don't know. There's too many tubes, right? The biochemical day, there are a boatload of tubes. But the little urea broths, most of the test tubes are pretty good size. These are littler tubes and kind of peach colored stuff. Is it inoculating this week, or are you have you already seen it? Inoculating this week? Okay. So the urea broth, there are some bacteria that can break down the urea. And if they do, you're going to get a color change in those tubes, and it'll kind of be pink colored, really, if it does its job. All right. The urea is sensitive and can't get cooked up in the autoclave. All right. It has to be filter sterilized. It's got to go through this syringe is really what they do with the filter sterilizing with it. Big ones like a 50 cc syringe. They screw the filter on and then they slowly squirt, I think 2 mils into each of those tubes, and boatloads of tubes because there's over 100 people in the intro lab, and I think 30 some odd people in the general lab. And you all do biochemical tests, all right? So things like that, you can't autoclave, and you have to use a different mechanism. And this idea, we'll get to the vacuum pump in 1 second. This membrane filter, there are a handful of different types of filters that we can utilize, all right? Some of them allowing bigger things through, and others much, much smaller, all right? So some may let a little bit of bacteria to move through those membranes. We're not thinking about that type of filtration, but you truly don't want that if you're trying to sterilize something. So there's another size, much smaller, that traps the bacteria. Virus could still get through there, but in these circumstances, we're not concerned about that stuff, all right? So this type of filtration, a very small filter pushing that fluid through. It's a similar idea over here, except this small little membrane filter that screws onto a syringe is now much larger, and we're thinking about big volumes that we could push through here. Sometimes you hear about boil orders for some towns where they've got some potential issues, coliform counts. So things like gut microbes, E. coli is a good one, that are potentially contaminating that water, right? I feel like it was Blackfoot just a couple of months ago that had a boil order. I could be wrong. So they do water testing all of the time, basically checking to ensure there aren't any issues. And they take samples of water and they run it through this filter apparatus. That filter is going to trap the bacteria. And after we have done that, we will take the filter and we will put it onto a Petri dish. And it basically inoculates that media with what we've trapped on the filter, potentially trapped. And if there are bacteria that are potentially disease causing, like the coliform bacteria, it shows up on those plates, right? Potentially you guys have worked with the EMB plates in the lab when you're looking for some gram negatives. They kind of give sort of a metallic-y green sheen on there. If you've got like a lactose fermenter, a coliform, that's what they do with water quality testing. And they sample it onto those EMB plates and see what's up. They use a vacuum pump, or potentially you've got a flask, a sidearm flask. You can hook that up to a sink. And as the water's turned on, it pulls air through the flask, and the filter that's up above, the water goes right through there. So you can be dumping your sample in, maybe a liter or so, and focus it on that membrane filter and inoculate it onto a plate. That's how they do things like that, water testing or other stuff, right? So either way, those are the ideas. When in C phages, when we're looking to find virus in like soil samples, we will take that soil and we will have it with some buffer, like the broth you'd grow bacteria in, but there's nothing, no bacteria in it. It's just the sterile media. And we allow that to kind of shake with the soil. If there are virus in there, as it gets kind of agitated, the idea is it kind of gets them up out of the soil, and then we centrifuge it and we take that liquid and we push it through a filter, and the virus would go through that. We mix that liquid with some bacteria, and if they can infect, when we do some plating, we see the little plaque formations on those types of media that we've inoculated it with. So handfuls of different things that we can be using these things for, and we can flip it around and use it to filter out the bacteria and hopefully find virus that we're hunting for, which I think is kind of cool, a cool mechanism to get there. So a couple of tables here to just kind of recap some of these ideas for control of microbes. And these are, again, physical means. So heat, numerous mechanisms from autoclave temperatures to maybe lower pasteurization temperatures, even higher heat as we think the incinerator, that, you know, your Bunsen burner or others. We talked freezing, and refrigeration is another mechanism that we think of as a control, but it's more of an inhibitory situation, slowing down the growth, all right? There are some organisms that we'll talk about a little later on that technically like these habitats and can grow and maybe release toxins, setting the stage for illness. So pressure, back to autoclave with this first bit, and then hyperbaric chambers, maybe thinking ourselves going in there. And wound treatment is a potential with that, especially thinking some anaerobes, because you've got levels of oxygen that you're really trying to infuse into that wound. We started this thinking like the, you know, dehydrating or salting things. So desiccation's the removal of water, so drying, and there's handfuls of ways. Freeze drying or lyophilization is a technique that allows for liquid to be removed very rapidly, all right? And a handful of other potentials. And then the radiation, ionizing and non-ionizing. I got one quick lyophilization story to tell you that really turned into kind of a nightmare. There was a professor that worked at ISU a super long time ago, all right? I believe he retired in 1980. That's a long time ago, right? So shortly before this, the pharmacy department got this new machine that could lyophilize things. It could freeze dry all sorts of things. And we still purchase lyophilized cultures of bacteria, little freeze dried cultures that you get. You take that little freeze dried pellet and put it into some broth, and it comes back to life. All right. And these lyophilized things could stay good for forever if you kept them dry, right? They technically could be all fine and good, all right? So we have all sorts of bacteria. You've already seen the hints of some of the bacteria that we work with. We've got quite a few, all right? And I would like to just stop for 1 second and think, you know, today what we work with is highly controlled, right? We know all of these things that we're working with, and we know some of these things that are potential pathogens, and you know, your potential to really work with those is limited, all right? But if we go back to the mid 1970s, right, there were all sorts of different organisms that we had the potential to work with, some of which were some true pathogens, all right, some serious disease causers, right? When you know, 9/11 happened, the Twin Towers came down, all sorts of terrorist sorts of ideas went through people's minds, and what sorts of things are people working with in labs, right? And that's when kind of there was more limitations put on stuff, right? Things that we maybe could have worked with, we no longer could, right? But this guy was working with all sorts of different things back then, right? And he had all sorts of cultures. And the pharmacy department gets this new lyophilizer. So he takes down all of these things, grows up lots of broth cultures, takes them down, and they lyophilize them. And they put them into these glass vials, and then they heat the top up and seal it shut. So now you've got these little ampules of white powder in these vials, and he retires. And they get forgotten about in a closet, in a cupboard, to be revisited decades later as this box gets pulled out. And there are all of these bottles in there that most of them just have some weird code, some numbers written on them. It doesn't say who they truly are. Nobody knows what any of this stuff is, right? All we can assume is it's bad news, right? Some sort of frozen pathogen, right? And that box gets moved to another cupboard and forgotten about for more years, right? And then another person retires and that box gets refound, right? And put on a shelf and forgotten about. And I'm just like, what? This isn't good. And then I decide we should really probably deal with these things, right? There were thoughts before that all of those vials should be opened and dumped into broth and allowed to grow, to put in the autoclave to kill, because that's what you got to do, right? So the thought was, but that's another big extreme hazard, right? And in turn, hazmat sort of situation was really where this stuff ended up happening. We don't know what it truly was, right? There are medical waste companies, they come and collect. You put all of your Petri dishes in those buckets, right? There are bags in those buckets that get pulled out and tied up, and they get put into a bigger bag in a bigger container, and those bigger containers get picked up and they get taken away, right? And that ultimately was the end plan for what needed to happen with this stuff. No longer our problem, right? Apparently, that stuff, I'm assuming, large autoclave that it ends up going to and then disposed of, but not here, right? Different, right? That's my lyophilization story. Finding this box of freeze-dried, white powdered bacteria that no one knew what it was, right? Definitely disturbing. And, you know, had it happened to go into, you know, just the autoclave or whatever, maybe all fine and good, but had it gone into someone else's hands, who knows what truly could have been growing in there. Definitely some issues, all right? All right, couple last little bits. Sonication uses sound, all right? Sonicating, it's almost like an immersion blender, but it's pulsating sound, and you typically have ear protection. The room you're working in, you put signs up so people don't just walk in there because your hearing can get damaged and you don't even know it. And then we talked the filtration. All right, I'm not going to bog you down with different chemical structures, but there are a few mechanisms with this, and I will mention the phenol. There are all sorts of phenolic substances, some of this stuff going into like the preservatives for the donors, the cadavers. It's really kind of a nasty substance. It is something that is in super low concentrations in the sore throat spray. Chloroseptic, I think is a name for that. That basically spray that in the back of your throat and it quickly numbs it and your sore throat feels a little bit better. And it's some of the phenol. You spit that stuff out after you let it sit there for a couple of seconds, right? This basically numbs, and high concentrations of it can be very dangerous, right? Biology building is pretty old, all right? We're in the talks of getting a new building. It's supposed to happen soon, but it will take years to build, so you'll be done, right? If you look at the main hallways, there are safety showers on every floor. There are showers, right? But if you take a closer look, where you see those showers, there are no drains in the floor. The safety showers were an add-on. It was an afterthought, all right? Because at the time, who needs that, right? We need that, right? There was a professor, oh, so many stories, not the same guy that made the lyophilized stuff. There was a professor carrying a 1-gallon jug of phenol. When you are transporting stuff that truly is a potential hazard, it's supposed to be in secondary containment. So that glass bottle should have been in a plastic container that was sealed. So if something happened, it would be contained in the other container, all right? That wasn't the case, all right? The bottle slipped out of his hand, hit the floor, shattered. He slipped and fell into the phenol, and it's concentrated phenol. So it numbed his skin He didn't know that his skin was being burned off of him. Not a good situation, right? So he gets kind of covered in this. At the time, there was a hospital right across the street from the biology building, so he was quickly shuttled across the street and cleansed, all right? And right after that is when all of the safety showers were retrofitted into the building, all right? That's why there aren't floor drains, because afterthought, all right? Crazy, all right? You want to be careful with stuff. I remember a chemistry class where the professor rolls in, and it's a TA teaching the class. So here rolls the professor. I'm like, what the hell's going on, right? White lab coat. He's got his goggles on. He's carrying a tub, and in the tub, it looks like a paint can with the lid sealed down in there. And he pulls the paint can out of the tub, puts it on the bench, opens it up, and in the can is a glass bottle, and in the bottle is a little hunk of sodium under mineral oil, because water would make it explode. And he pitched some into some water, and we watched it explode. It was pretty awesome, right? But that was my first hint of like secondary containment, seeing this little bottle in a can in a tub. Crazy, right? And had he dropped it, the idea is it stays in this can and there's no water in there, so you're not going to have an explosion. That's the idea, right? All right, enough of that. So this triclosan, I question, can you find this? It's something that I'm pretty sure has been banned, and we will leave it at that. It's something that definitely is an issue. There's all sorts of ideas with metal that might be antimicrobial: copper door handles, silver, stainless steel on the inside of this incubator, antiseptics with maybe some zinc in there, mouthwash that is antimicrobial. And this poor individual over here, I mean, is he trying to be in the Blue Man Group? I don't know. Is he, you know, I don't know what's up. Colloidal silver is something that some individuals feel harbors all sorts of good potentials and may make them healthy. This is a bioaccumulation. So it's not like he just took one little dose of colloidal silver and turned himself blue. He's probably been consuming this stuff for decades, and in turn looks like this, all right? My sister informed me that she has a neighbor, an elderly neighbor, similar blue looking dude, right? I'm like, it's probably this guy, right? Colloidal silver, topical applications, maybe okay, all right? But to consume it, it does ultimately accumulate within your body, and this is irreversible, blue forever. That's pretty crazy. Here's some iodine. We think of this as a surgery prep topical antiseptic, and we talked a little bit about that previously. So some alcohol, another potential ethyl or isopropyl, and that might be something that we could think about for preps as well. If we were going in and not, gonna maybe do a spinal tap or something like that over in this guy. If we had if we were going to donate blood or give a blood sample, a quick alcohol swab would be what would be utilized with that. So soaps, another potential with this, and I feel like soap is effective by itself. Throwing a bunch of antimicrobial agents into your soap truly sets the stage for resistance for microbes that may not potentially get cleansed away, or we see the tolerance within them building, and it sets the stage for bigger problems. one of those mechanisms why the triclosan was discontinued in the first place. Lots of organisms started developing resistance to that. Bad news, all right? So just the simple action of soap is enough to break down the kind of lipid layer in your on your body, and the transient microbes, the potential pathogens, they're on the surface of that. So the soap gets in, breaks that layer down, washes it away, and underneath are your good guys, right? You're never going to be able to cleanse everything away. You technically won't, those good guys underneath, right? But soap helps to remove the bad guys in that oil layer, making it easier to rid yourselves of those possible pathogens. So similar idea with that. And hand washing, I hope we all are pretty competent with these techniques. And I believe at the Starter Lab, you did a little bit of hand washing stuff to see some of that. I remember some of these labs when we would do some of this hand washing, and you know, we've changed stuff a little bit over the course of semesters with how we do our plating and all of these things. But we'd start off with wetting our finger, touching the plate, and then a little bit of washing, then throw some soap in, and then wash for about 5 minutes or so, kind of like thinking a surgical scrub in, and seeing what happens over the course of each of these different types of washing procedures. And the very last one, when you really did some lengthy scrubbing, you oftentimes saw a lot of growth on the plate compared to maybe the first one. And people would maybe get the wrong idea and think, holy cow, I'll never wash my hands again. Look at all of these microbes, right? But it's not microbes. It's really, look at this microbe, right? It's like one type of bacteria. And that initial first touch may have had 8 or 9 different kinds of bacteria, a lot of variety, a lot of potential pathogens that you have picked up just by touching things. It's different, right? The growth may have been a little less, but it's those good guys that really shine through after that washing that lets you know it is truly effective, right? And we don't think of them as really being culprits for disease in the first place. All right, handfuls of other agents. Some of these, like I mentioned, those Petri dishes, that was maybe last time, but the plastic dishes that they pour the media into that you inoculate all sorts of bacteria with, they sterilize those with ethylene oxide. So they pipe that gas in, and it in turn makes it so that that plastic, those dishes are sterile when we open up the pack and start pouring the media into them. And handfuls of other things. Catalase activity, this is another thing you'll see with your biochemical tests soon. Some bacteria have the ability to break down peroxide, others don't, right? If they've got the enzyme catalase, peroxide gets converted into water and oxygen gas. And you see the little bubbles as a result of that. We sometimes put it on, you know, you get a wound or something like that, and you see some bubbling action. And really, it, you know, your blood, there's some catalase within that. Blood will bubble on its own. But if there are some bacteria and we see the bubbling, you're really not killing those bacteria. Those bacteria are eating the peroxide, really. They're not eating it, but they're breaking it down, all right? The bubbling action helps to cleanse, to bring the stuff up and out of that wound. But the bubbling doesn't mean we've killed stuff, all right? But what about the opposite? What if there was no bubbling? Maybe that's a different story, right? Those microbes might not tolerate it, right? And in turn, maybe that's when we really see it being effective. There are lots of different things that may have catalase activity, but when we really think about this in a biochemical situation, it's oftentimes the gram-positive cocci that we think about. Staphylococcus has catalase activity and Streptococcus doesn't. And so a simple drop of peroxide on there would show us bubbles or not. And there you go. All right. And with that, I think that's pretty much it. We will pick up just the last little tidbit and start the next chapter on Friday. Some of these, I think, you might get a chance to see in the lab with like the susceptibility. All right, farewell. Good day. So close to finishing.